My name is Spencer Schiavo, and I, along with my partner Nick Agunzo, am studying the effects of soil pH on the living environment across Long Island. Before we can study the effects of acidic soil, we must learn how the acidity in soil across Long Island is derived. Primarily, acidic soil comes from the precipitation of acid rain, which forms in the atmosphere. Specifically, pollutant molecules such as particles of sulfuric and nitric acid become sources of condensation nuclei, and thus water vapor condenses to these particles and become an acidic solution. The environment's ability to combat the rapid decrease in soil pH, also known as its ANC, or acid neutralizing capacity, depends on the geological composition of that environment. The chemical properties of the types of rock within the soil plays a factor in neutralizing the acid. A process known as cation exchange is a specific chemical reaction that makes acidic soil detrimental to the plant life of the environment in that H+ ions within the soil are exchanged with basic ions in the surrounding humus and clay particles, leaching key nutrients. Also, soil acidity, as a function of depth, directly increases. But how does this affect the environments on Long Island? Acid rain is present across the eastern U.S., especially Long Island. Soil acidification creates a constant pressure on the organisms within the environment, disrupting equilibrium. Finally, pollutants have a strong presence in the eastern United States, which is a main cause of the formation of acid rain, as explained earlier. Although acid rain is a primary component in the formation of acidic soil, the geologic composition of the affected area is a key determinant in the environment's ability to adapt to acidity. Long Island, specifically southern Long Island, is comprised of a sandy quartz bedrock structure as a result of a receding glacier that left sand deposits. Sandy quartz has no basic properties to neutralize the acid, therefore letting the soil acidity cause catastrophic stress to these environments. The basis and conducting of our research is in Heckscher State Park, a state-funded area located in southern Long Island. The first component of our research is to collect the soil from this site and determine the relationship between soil depth and soil pH, as well as establish a general idea about the soil conditions existing on southern Long Island. Furthermore, an analysis of plant species within Heckscher Park will determine a correlation between the soil conditions and the plant's living conditions. Then, running future experiments involving specific plant species found at Heckscher will aid in determining a common link between soil and optimal plant growth conditions. Dividing Heckscher Park into quadrants allows for easier organization and ensures an unbiased sampling of data. This diagram shows the division of Heckscher State Park into 3 quadrants. Within each quadrant, 2 sections, shown in red, are marked and recorded by GPS coordinates. Within these sections, soil collection and plant analysis take place. For observing the plant species, we need 3 materials: leaf snap, an iPhone app, plastic bags, and tape or string and a GPS. First, mark off each 25 by 25 foot section using GPS to mark the coordinates. Then, collect leaves of each species within the section and put into plastic bags. Also, observe the frequency of each species. Finally, using the iPhone app, Leaf Snap, take a picture of the leaf, and this will find a match of the specific plant species that leaf pertains to. For collecting soil, we need 6 materials: an auger, small shovel, plastic bags, paper, GPS, and tape measure. First, use the GPS and recorded coordinates to find each section. Then, we dig in depths and intervals: 0, 10, 30, 50, 70, and finally 90 centimeters. But use the small shovel for collection of topsoil and 10 centimeters. Ensure accurate depths using a tape measure. Then, for deeper holes, use the auger. The auger length equals 20 centimeters, therefore collect soil from the bottom of the auger to ensure accuracy. Tap soil from the bottom of the auger into the sheet of paper and slide each sample from the soil in each interval into a separate, pre-labeled plastic bag. For the soil pH testing method, we need 9 materials: 6 plastic test tubes of at least 100 milliliters, 0.01 molar calcium chloride solution, 10 milliliter beaker, 2 millimeter sifter, paper towels, sonicator bath, a beaker of at least 500 milliliters capacity, styrofoam test tube holder, and a pH meter along with its respective probe. First, pour each sample of soil onto dry paper towels and let them sit for at least 24 hours for drying. Put each dry soil sample into new, separate, pre-labeled plastic bags. Sift each sample using the 2 millimeter sifter, Pour 10 milliliters of the soil from each sifted sample into the pre-labeled plastic test tubes. Measure 10 milliliters of calcium chloride 2 solution using the 10 milliliter beaker. Pour into each test tube filled along with the 10 milliliters of soil. Then, mix the soil calcium chloride 2 solution together in each test tube. Place a set of 3 test tubes in the Sondigator bath. Run it for at least 5 minutes. Repeat for second set of 3. Store the 6 test tubes in the styrofoam test tube holder and store for 24 hours. Calibrate the pH meter and carefully place the probe in each test tube to determine the pH. Thus far, here is our recorded data. Plant species that were observed at each section are recorded along with their respective number of occurrences. The recorded pH of the soil as a function of depth is also recorded here. At first, our results were slightly unexpected and likely inaccurate due to possible experimental error. However, as our experimental procedure regarding the testing of soil pH became more efficient, our results became more in accordance to our hypothesis. A general trend also started to develop in that soil pH as a function of time increased as expected. Analysis of plant species existing at Heckscher State Park lead to further research into the optimal growing conditions of each plant species. Essentially, every plant that was observed is optimized for growing in sandy, acidic soils, although some of the optimal pHs range from species to species. This establishes a relationship between the soil conditions and the plant species in that these organisms are generally well adapted to the acidic environment. However, it is still unclear if these species emerged as a result of natural selection within the environment or were already pre-existing. Further research will allow us to determine a plausible explanation.